Welcome to Genomics Now, a podcast series where you can learn how genomics is developing in England's NHS. This podcast series is recorded in 2024 and is part of the North Thames Genomic Medicine Services Educational Toolkit. In each bite-sized episode, we explore what is changing in genomics. We answer the questions on genomics we've been asked by the different healthcare professionals. We talk about the ways genomics may impact different healthcare roles, and how genomics can be used to successfully improve patient care. Hello, I am Dr. Shazia Mahamdallie, and I'm part of the North Thames Genomic Medicine Services Workforce Education and Training team. Today, I have the pleasure of chatting with Professor Mike Hubank. Mike is the head of the Clinical Genomics Pathology Service at the Royal Marsden NHS Trust here in London, and he also serves as the Scientific Director of the North Thames Genomics Laboratory Hub. In today's conversation, we're diving into an intriguing topic, the process of transferring technology into NHS laboratories. We'll specifically explore the technical evidence supporting the pilot of liquid biopsy circulating tumour DNA testing. So stay tuned as we unpack this exciting area of genomics. Welcome, Mike.
Hi, hello, Shazia. Lovely to be here.
It's great to have you here. So, Mike, can we start with some basics? We often hear about advanced genomic technologies, how becoming part of testing and how they're becoming part of testing in pathology. Could you explain what that means and how liquid biopsies and circulating tumour are examples of advanced tech in helping to progress lung cancer testing?
Yes. So in the past, there were limited ways of doing molecular testing. They worked, but you had to know what you were looking for. So in techniques like FISH and PCR, you had a specific, very focused target that you had to look for. And then if that test was negative, you then have to guess another target and guess another target. The problem with that is that while it can give you the result if you get it right, if you get it wrong, you don't know what to do next. Now, in parallel, some great advances were made with next generation sequencing, high throughput sequencing, which really brought the price of that down. What that's allowed us to do, instead of targeting a single variant in a gene, we can look at the whole gene. We can look at multiple genes at the same time. So instead of doing sequential testing, we're able to do all the tests we want in one go, so we don't have to guess at the target. That's had a lot of impact in terms of what we're able to find, because we can see lots of different types of variant, like single nucleotide variants, copy number variants, all of these things we can pick up at the same time. We can look at DNA and we can look at RNA, which allows us to look for gene fusions, again, usually done by FISH in the past. And this also allows us to look for new targets as well that could be perhaps useful for clinical trials or research. So we don't have to limit to what we already know is testable. Now, where that fits in terms of circulating DNA markers, that's really an extension of the technology, going from looking at the tissues to looking at blood. And that's been a very exciting development over the last few years.
So it's kind of like a question of you've got that extra added scale as well as a different way of different material in which you can actually do the testing on. So you've mentioned the new tech and it's opened up those new opportunities. You recently brought liquid biopsy testing into your lab through a tech transfer. Can you tell us what that involved and why it was such a significant move for patients and your team?
Yes, absolutely. So, circulating biomarkers is really the next frontier in diagnostics. So they led us to make decisions on patients where taking a sample is very difficult, dangerous, or sometimes impossible. It's a more expensive test, the liquid biopsy, and there are some limitations with it in terms of interpreting a result where we don't get an actionable variant. But on the whole, it can be faster, it can be much more efficient, it can still be comprehensive, and it can save lives. And of course, you're taking this from a blood sample, which is very, very easy to access. So the motivation was very clear on why we wanted to do this. The challenge was really about sensitivity. So sensitivity really is like looking for a needle in a haystack. Most of the DNA circulating in the blood is not from tumours, it's from white blood cells or for endothelial, the blood lining. So what we're trying to find is just the bits of DNA that come out of the tumour, and that requires us to have a very, very sensitive test. And you have to be more sensitive than the errors that the test makes, if you see what I mean. And in order to get around that problem, we work with a company called Guardant Health, who put a lot of money, well over $1 billion, into developing and validating a test. And while we could try and replicate that, it seemed to make a lot more sense for us to partner with them. But we did want it to be a partnership. We didn't want to just buy a service. We wanted to have control over how we ran this test, what we did with the data, how we reported the results. So we spent about 2 years on working with this company to set this up in house. And I look at it a bit like a bit like the Kentucky Fried Chicken of genomics. We kind of do all the work, but it's their special sauce, if you like, that makes the results taste so good. And that's given us a lot of benefits in terms of robustness and sensitivity to report and find the variants we need for patients. And I think, in the long run, we'll be looking to do more of this kind of thing.
Yeah, no, that's a really great collaboration. And at the end of the day, like you say, it has that patient focus at the end, something that's less invasive and has much potential for them. So I'm keen to go a little bit behind the scenes of your laboratory. So could you walk us through how liquid biopsy testing works in your specialist path lab?
Yes, absolutely. So, Shazia, we've been part of the lung transformation project. So this is a clinician-driven but NHS England supported project for looking at rapid diagnostics through circulating biomarker testing in lung cancer. We've used the platform, we call it the Marsden 360 platform, which is the test that we run in collaboration with Garden to do this. And the way it works is that we'd receive blood samples from doctors who've sent them taken from the patients. They send them directly as soon as there's a likely presence of lung cancer. So as soon as there's a diagnostic scan or anything like that, instead of sending the tissue as well, but primarily they'll send us a blood sample. That blood sample is just a simple bleed and send in it, to us in a tube, and we will receive that directly in the lab. It doesn't go through path labs, comes straight to us, and we will then sequence that and try to return those results ideally in 10 days or under. I think we're currently a little bit over that, but we're working on capacity because it's been extremely popular. So we're running literally hundreds of these tests a month, and it's comprehensive. And the great thing is that we're able to issue our own reports in line with UK recommended treatment pathways, rather than trying to have a global sort of generalized approach. And that results go straight back to the clinicians. We've had many examples already of actions that clinicians have been able to take where a patient previously would have not been well enough. And that's simply a function of time. Lung cancer progresses very quickly. So getting those circulating biomarker results back quickly means patients can get onto a treatment that they otherwise might not have managed. And we plan to increase this process as we go along.
Oh, amazing. So you mentioned there that you've got that partnership between the NHS, Garden Health and yourselves, and it's currently in pilot phase. Doctors are sending samples to you. So, but from your perspective, from a clinical genomics pathology service, what data do you need or would you need to ensure that liquid biopsy testing becomes actually something reliable and an option for clinical use in the future?
Yeah, it's a very good question. So because it is a little bit more expensive to run these tests than the standard tests, we have to demonstrate value for money, I think, across this. It's not about technical equivalence. We know now that circulating biomarkers will give you very similar results to tissue. Tissue and blood testing is slightly different. So tissue testing can sometimes be a tiny bit more sensitive in some areas, and then blood testing can be more comprehensive because it finds variants from bits of sample that you haven't sampled, if you like, bit of the tumour that you haven't sampled. So technologically, we know that that's there. The issues really now are to do with how best to implement the methodology, how to ensure value for money. And so we want to deliver the right test for the right situation. Every different indication, every different cancer type has got perhaps a different requirement in terms of what we need to find and what's the best way of doing that. Now, in order to do that with the right sensitivity and specificity, we need to tailor these tests to the needs of the individual patient. It needs also to be affordable and deliverable within the NHS. So we've actually formed what we call a circulating biomarkers network of excellence supported by NHS England, which is doing just that. So we're looking at everything we might need in order to get those correct tests to the right patient at the right time.
You said there that there is potentially to go to other tests and other indications. It's the right person at the right time for the right indication. So just wrapping up, we're Genomics Now, and we're in a pilot phase for this. I'm curious about your thoughts on what's next for liquid biopsy testing. Do you think there's potential to scale across the service across England, extend beyond lung cancer? What do you think would be needed to make that necessary?
Yes, I really do. I mean, I think the future of liquid biopsy is very bright. I can see this increasing. It, what's not to like about it? It's a blood-based cancer test, and we do always have to bear in mind that there are some limitations. It's not going to completely replace tissue testing, although it may do in some circumstances. there's a very clear need in lung cancer. And I think we're using that as the kind of the pilot vanguard study, which will demonstrate, real benefits and utility. But beyond that, part of what we're doing with the biomarkers network of excellence is to look at other indications. Cancer of unknown primary is a very good one, where you don't know what the tumor type is. Sometimes it's hard to sample. And again, we can pick up indications, signatures of what cancer it is from sequencing circulating biomarkers. Similarly, in advanced breast cancer, you have circumstance where the cancer has changed. It's developed a new resistance methodology. It's developed new drivers like ESR1. Very hard to sample advanced breast cancer. So circulating biomarkers clearly seem to be the way to go with that. Pediatric cancers, again, you don't really want to be taking a lot of tissue from young patients when you can take a blood sample much more easily. And there's many others, pancreatic and biliary cancers. There is now building evidence that we can do this, germ cell tumors. But each of these things is going to need a different approach, a tailored approach. Gives us the best value for money in the NHS, but clinically benefits that patient. So there's a lot of work to be done, but that work is going to pay dividends in the long run, with better testing, faster turnaround results, leading to more impressive and hopefully safer results for patients.
That's really interesting, because you raise a point there that is not only about different tumours, but actually how a tumour can evolve. And as it's a less invasive test, being able to understand that evolution and then the treatments that could change as somebody's cancer changes is actually a coup for this type of testing.
Absolutely. And we want to move it on to other technology, other applications like monitoring residual disease. So to see if the cancer is coming back. If we can get a test cheap enough, we could do that at a regular interval from a blood test, pick that result, pick an indication that the cancer is coming back up as early as possible, so that you can then change treatment, anticipate what's coming back and get in there and do it. There are also applications for looking at treatment for cancer vaccines. So there are many different applications that we've got for circulating biomarkers. We've got the technologies. It's really a question now of working out what's best in which circumstances. And I think, there'll be a lot of work in that area over the next few years.
Yeah, a bright future, as you say. So thank you, Mike. I've really enjoyed this very short conversation, but really informative conversation. And I'm looking forward to seeing how liquid biopsy testing develops in the NHS over time. As you say, there's a lot of potential there.
Well, thank you very much, Shazia, for inviting me on. It's been lovely, and I hope it gives some indication to people of the exciting area of circulating biomarkers.
Always a pleasure. So,
thank you for listening to this episode of Genomics Now, a series of conversations hosted by healthcare professionals. The North Thames Genomic Medicine Service aims to complement other genomic-focused educational tools with these bite-sized podcasts. So do visit our website for more resources and updates on genomic-based clinical developments and research. If you've enjoyed this episode, don't forget to share your thoughts and spread the word by tagging the North Thames Genomic Medicine Service on social media.